Thank you very much for that kind introduction, and thank you for asking me to speak today. I'm going to start by showing you a schoolboy's examination paper, because the triangle of Pythagoras takes us right to the start of the beginning of the understanding of blood. Pythagoras wasn't just a mathematician, he was also a physician and advised his followers not to eat fava beans. He didn't know at the time why not to, but he knew it made them very sick. And Joel Newman, the last speaker, will explain to you why at the end of these three talks. And Deepti Radia, my colleague in the centre, will talk about the present. Joel's going to talk about the future. So I'm going to talk about the past. So here he is. Pythagoras realised that his followers got very sick and became very pale. And of course the ancients knew very well about the heat of the blood and the spirit of the blood, but they didn't understand the science of the blood. And there was a huge long gap between Pythagoras and then the Renaissance anatomists and then the Enlightenment scientists who began to explain more clearly what was happening in the blood. And Zacharias Janssen doesn't get enough credit for inventing the microscope. He was one of the first people, I think, to use a compound telescope. And there is a rumour with no foundation, as far as I can tell, that he may have learned this from Islamic culture. So Janssen was actually the first microscopist, I think, that we know of. And he showed Leeuwenhoek his microscope, and Leeuwenhoek copied it. And this is all that's left of our understanding of the structure of Leeuwenhoek's microscope. Goodness knows how anybody saw any microscopic images down this. And Leeuwenhoek never told anybody how he illuminated his objects. It remained a secret to the grave. Here he is. And here is the man who took Leeuwenhoek's findings of blood cells further. Leeuwenhoek was the first to describe what blood cells looked like under the microscope, but Hooke from the north of England was the first to begin to describe in detail the structure of blood cells. I don't know if you can see this clearly, but Leeuwenhoek made a mistake, which was that red blood cells were flat discs. Hooke made an extremely important observation. That was incorrect. They were in fact biconcave discs. And in the 19th century, a century later than Hooke, it became clear that was because that was the way to allow most of the molecules of hemoglobin access to the cell surface so that they could exchange oxygen efficiently. If it was a sphere, too many hemoglobin molecules would be at the centre and couldn't transmit oxygen, but this arrangement of the cell made that possible. And Hooke also discovered blood clotting. Here are red blood cells caught in a web of fibrin with some little cells which he called blood dust. You can see the little blobs, which are in fact what we call platelets now. So Hooke is what we recognise, at least in the UK, as the father of haematology. And here is what we use now as shiny new transformations of the microscope with which we're familiar. Then Virchow came along and made a lot of important observations, but in particular, he began to describe the cells that you see in patients with leukemia. He recognised the difference between normal white cells and abnormal white cells and their relationship to red cells. And this blood film simply shows you a few red cells and one white cell. And then after Virchow's discoveries in the late 19th and early 20th century, we began to realise that there wasn't just blood, there were different types of blood. And that was the beginning of the history of blood transfusion. The realisation that you couldn't just give any pint of blood to anybody, that you had to do very careful cross-matching. And that science has progressed since then. Now, instead of plain microscopy, we have cell counters, which Deepti will explain to you in more detail later, so that we can process large numbers of samples of blood very quickly with very cheerful laboratory technicians doing the work for us. And now we've got to the stage of very advanced scanning electron microscopy. So here's a white cell eating bugs on the surface of a blood vessel. So that we're getting further and further towards the present and the future now in this brief talk. And this is one of my heroes in particular. This is Sidney Farber, who was the first doctor to treat children with acute leukemia. Having understood the nature of white cells, having grasped the history of the distinction between normal and abnormal white cells, Farber was the first man to treat children with leukemia successfully. And it's been part of my career I'm most fortunate to have enjoyed, and of which I'm proudest, to show that we can actually improve survival of patients with acute leukemia, even in adults. These are what you call actuarial survivals, which your insurance company constructs on your chances of being alive at certain time intervals. The bottom curve is where I started in my career, and the top curve is getting close to where it is now. So we say at 5 years from diagnosis, at least 50% of adults with leukemia under the age of 60 are survivors, and that probably means they're cured. So that's a massive advance in a very short space of time. And one of the reasons we can do that is because we can now split blood into its various components. One particular one is platelets, the little cells that Houston discovered causing the blood to clot. And if we don't do that, our patients with acute leukemia who are given intensive chemotherapy do not survive because they bleed to death. Platelet concentrate's an extremely important component of blood. We're now also able to separate stem cells from peripheral blood and concentrate them in bags to give stem cell donations for patients to have transplants from relatives and from people who are not related but have tissue matches. We're also able to make a huge range of diagnoses now on blood spots taken from the heels of children. This is what's called a Guthrie card, and it can be subjected to very detailed molecular investigation now for a number of inherited disorders. Here is the DNA double helix with all its little nucleic acids, and here is the code of the DNA called the genome, which we now know can be investigated very thoroughly. And this is not an up-to-date list of diseases which are known to have single abnormal genes and therefore present targets for treatment. This list grows every day and is part of what's called stratified medicine or targeted medicine. So we're moving away from an old-fashioned descriptive type of understanding of disease, beginning with Pythagoras, now to very exact molecular diagnoses simply by studying the blood. And here's an example of a patient with melanoma. It's not a blood disorder, but it illustrates what I've just been showing you, that if you find a target on a tumour, however extensive the tumour might be, and you produce an antidote or an antibody to that tumour marker, you can wipe out a huge amount of tumour with one single molecule. And to close my section, I think the most exciting history is how we've managed to treat patients with haemophilia, an extremely nasty bleeding disorder in which the factor VIII or the factor IX gene is missing. So patients don't make a key clotting factor. Well, now we can see where the gene for the factor VIII and the factor IX molecules lie. And we can snip out that gene, we can insert it in the haemophilia patient's white cells, and we can stick the white cells back into the patient, and they start to make the missing factor. Real gene therapy. And I'm pleased to tell you the first-in-man experiments to show this worked were performed in London by one of my colleagues, Professor Nathwani from UCL, who's shown that in Christmas disease, you can in fact make this molecule reappear that's missing from patients with the disease. So from Pythagoras to gene therapy in 3 millennia is a rather fast leap for an introductory talk, but it sets the scene for Dr. Radia.
Thank you very much. Dr. Prentice has eloquently described the 3 millennia in a very brief time. As Professor Hooke said, this lecture series is based on objects and 50 objects. I'm a consultant and I work at Guy's and St Thomas' Hospital, and my remit was really to choose 3 objects which I felt made a huge difference in diagnosis and where we are today, what I'd work with in the laboratory or in the lab, which I take for granted. And I'm going to speak through the microscope, the Coulter counter and the glass slide, because those are really the mainstay of what I use in terms of everyday diagnostic work. So we've had lots of disputes about who is the father of microscopy, and it'll depend on whether you're Danish, whether you're English, and where you're from. But as Dr. Prentice has already pointed out, the first documented compound microscope with 2 lenses was really produced by Zachariah Jensen, who was an optician, putting 2 lenses together and realizing that actually you could magnify objects to a certain degree. But it's not been new. Glass was invented in the 1st century. The Romans were experimenting with it, and they looked through glass and realized that looking at objects through glass, they were magnified. In the 13th century, spectacle makers made monocles. They were really glorified magnifying glasses, which then allowed for objects to be viewed to be slightly larger. And in the 1590s, I think it was, where Jensen actually produced the first compound microscope. But when did it transfer over to where I am now as a physician or scientist having a look? So it was really when microscopy became a part of what you did in science to look at objects that were not possible to be seen easily by the naked eye. And Robert Hooke, who's a British UK scientist trained in Oxford, and actually published the first book, Micrographia, which he was most well known for. And he actually examined fleas and insects under the microscope and illustrated his own diagrams. And the two famous ones were those of a flea being able to actually visualize the hairs that were not visible to the naked eye, and also a sliver of cork where he described pores and cells. And it was the first time those sort of words were used. I've been reminded that actually there are no official photographs of Robert Hooke, and he has an association with this college, so I'm not sure where that's come from, but it definitely wasn't under the microscope. We've also had mention of Anton van Leeuwenhoek, who was a Dutch draper. He had no scientific background whatsoever, and Dr. Prentice has also mentioned the very small microscope that he developed. That instrument there is no more than 3 or 4 inches long. It has a single lens that was based there and a screw that moved the object across the lens. And what he initially used it for was to look at the weave of fine cloth, to look at the number of threads in that fine cloth. But he was a bit of a philanthropist and was very interested in how objects or how this instrument could be used. He was credited as being the first person to describe blood cells or bacteria under that microscope. He was also the first person to credit, and as Dr. Prentice said, no one quite knows how he illuminated these particular objects. We know he wasn't his own illustrator, unlike Robert Hooke, so he had somebody there illustrating for him. And his expertise was actually, he ground up to 500 different types of cells to make the magnification much higher than it had ever been before. So the magnifying glasses used to magnify things up to 5 or 6 times. With his particular instrument, it was reported that he could magnify objects up to 270 times at that time. So that was the highest magnification. And so he started looking at drops of pond water, blood, and he started writing letters, and he corresponded extensively with the Royal College. And initially, when he was the first person to describe bacteria and single cells, his credibility was doubted. So the Royal College sent a team of observers to come and see his work, and validated his work, and he then became a fellow of the Royal College. So that developed, both were fathers of microscopy in different ways, and that developed a whole field of science where we were able to look at diseases at a microscopical level. And in the background, that's the microscope developing from a single monocular microscope to now looking a little bit more like the binocular microscopes that we see. So it was really a century and a half of image analysis, lots of men, lots of technology, moving us to the microscope that we see today. Ernst Abbe in the late 1800s worked with Zeiss, a German company, to try and refine the optical images. So you had microscopes that were used, they became binocular, the images weren't so great, and then the lenses started to be improved so you could get variable magnification. And so that's today's microscope. Our ancestors wouldn't recognise these at all, varying in size from the electron microscope. We now have wonderful IT and PC. We see the images on our computers or perhaps on a visualisation above the microscope, and we can take photographs of our images, so we don't need illustrators. And if anyone draws like I do, a photograph is much better. This is really a workstation of my histopathology colleague, who's very tidy. But this is his mainstay, so the microscope and the PC that he's got. And this is our teaching microscope. Sometimes 2 eyes are better than 1, or multiple eyes when we want to discuss and teach as well. But actually, we've moved on to the era of virtual microscopes and scanners. And this is the reality of today. This is what we're using, where your glass slide can be scanned onto the PC. And that slide, this at the moment doesn't happen to be blood, but a skin sample, could be used like a microscope. So you can actually move that image across on a PC. All those images are stored permanently, so unlike glass slides, they don't get broken or wear away with time. And they can be held in an internet or a host website, so they're there forever. And they can be used, and they are currently being used to make diagnoses. They can be used internationally when somebody can access the host site, and currently we're using them for education as well. So moving from that single small microscope to where we are today is a huge leap, but it's a fantastic progress to where we are. And this is really just a simulated mock-up of what can be seen in the quality of the virtual screen. It's only a snapshot, but the magnification can go up on your PC and you can move the slide along as well. So a really quick march through microscopes, but without a microscope I can't imagine how I do my daily work. Blood sampling, well, nothing's changed about taking blood samples. We still use the same veins. What's changed is we don't use IV syringes. We don't use a little glass bottle with the specific wooden carrier for it. We've got vacutainers. We have a whole host of blood sampling bottles with various media because we have a huge repertoire of blood tests. So your one single drop of blood goes a long way. And this is through all pathology specimens. And you have to forgive me, as a haematologist, I'm going to focus on haematology. The glass slide, I chose the glass slide. The glass slide really is where all of us as pathologists and haematologists fix our material that we're going to view. And a small drop of blood is put at the end of a glass slide and smeared across. That's fixed by particular stains, which bring out the morphological features that we look at to decide what's going on with your blood. And if the window is the eye to the soul, your blood is a barometer to your well-being. And we can get a lot just from a full blood count. So we now have automated stainers that actually stain the slide in a short spell of time. But the glass slide, looking back in the Victorian era when the microscope still had a stage, was really a small bit of glass mounted on two bits of ivory, which slid across at the objective to be seen. I understand in the late 1800s, the Royal Society of Microbiologists created slides that were standard dimensions, so actually the visualization was ideal, and we haven't changed those dimensions. We now have glass or perspex slides. So the blood film tells you a lot, and the full blood count is very, very informative. It's one of the most common tests done. And if you spun your purple top of full blood count, 45% of it is made of these red cells. A very small percentage is made of white blood cells and platelets, but they're very vital cells, and 55% of it is plasma. This is a blood film photograph, is what we'd see under the microscope, and this is an electron micrograph of the cells. So I'm going to pause there because before Wallace Coulter, who was an American inventor and scientist, came across the Coulter principle, all the full blood count was manually counted. The cells were looked at, they were differentiated, they were manually counted. So actually getting a full blood count with all the different cell types could take up to half an hour, 45 minutes, the components. But Wallace Coulter was an American inventor in 1953, discovered the Coulter principle, where cells could, in a fluid medium going through a small aperture, could be distinguished by the size of the cell and the light scattered through that aperture and the electrical impedance. So they could be sorted essentially in terms of their shapes and sizes, which was fantastic because he applied it to blood and developed the Coulter counter, which automatically counted those blood cells for you in a very short spell of time. And again, like the microscope, I can't imagine life in the lab without a Coulter counter. I can't imagine sitting there doing the differential. We do it for various disorders. But so he revolutionized the way that full blood counts were done. And he had 85 patents of lots of things, and this is the one that made a huge difference for the most common blood test that's carried out. So this is our lab at the moment. We've got the microscope with a PC where we can actually generate your purple tube, which is your full blood count, and our equivalent to a Coulter machine. Of all the automated Coulter counters, 95% use the Coulter principle. And what happens now is a small tube is placed here at the aperture, a small drop of blood. Within 2 minutes, you have your full blood count with all its differential. And that's very important and informative. So at Guy's and St Thomas's, we actually process 2,000 samples a day, of which 10% are made into blood films, which we have to look at under the microscope. So this has made us be able to turn around the work a lot quicker and get back to actually what we're here to do is diagnose the abnormalities, get back to the patient and make sure that we make the right diagnosis fairly quickly. So really those three objects are still the mainstay. Where are we? Well, the future's small. The future is really here. If I now have a very complex case and I'm concerned, I'm not doing that on my own, I can contact my colleagues overseas, I can scan the slide in, They can access those slides, and we can have a face-to-face conversation about them. We have lots of multidisciplinary meetings regardless to make sure that we've got the right diagnosis, and all this technology, which is useful that we can harness, allows us to make the diagnosis more specifically quickly and allows us to have the workforce which can work 24/7, providing the IT works. So those three objects at the bottom have now translated to where we are today. I'm going to stop there and hand over to my colleague Joel Newman.
Good afternoon. My name's Joel Newman. I'm a registrar working on the sunny south coast in Eastbourne. It wasn't so sunny there this morning, though. And I'm going to talk now about how far things have come since the invention of the glass slide and the microscope, which of course we still use. But that now just forms the basis of what we do in day-to-day life. As members of the general public, I'm sure you're more aware than I am of where we can take blood samples. General practice, A&E, inpatient department, outpatient department, boots the chemist these days. Everywhere takes blood samples, and they all come to a central laboratory for further investigation. But there are many thousands of different blood tests. Some people come in and say to me, I don't know what's wrong with me, do every test you can. Well, it might be a bit pricey to do that, but actually you probably wouldn't have enough blood in your system to do all the different tests that we have available. So when anyone comes to see us saying, I've got a problem, the blood tests that are selected are based on what the concerns are. And then we can work out from there, so as not to drain you dry. So we're going to join a few hypothetical patients today, just seeing what journey their blood tests make. Our first patient is James. He's 35 years old. He works in the city, so not too far from here. He works all the hours under the sun, and he gets very tired. He puts that down to being a hard worker. But now he's getting even more tired. He's having to stop at the top of the stairs to catch his breath. And he's also found this lump in his tummy, and he's not sure what that is. So he goes and sees his GP. And the GP sends him in to the Medical Assessment Unit, or MAU. If you watch television, it's sometimes called the AAU on Holby City. It's not that glamorous, unfortunately. So they do some blood tests there to look for anemia as a possible cause of his breathlessness, and to see what's going on. We also do that archaic thing called a blood film on a glass slide. But that forms the basis. That used to be where things stopped. His blood test showed that he was very anemic, with roughly half the red cells he should have. And on here, all these things which look purple, these are actually white cells. They've been stained up. And you can normally see these sorts of white cells in the bloodstream of a normal person. But these larger cells should not be there. And this is characteristic for a condition called chronic myeloid leukemia, or CML. Now that would be where it stopped in the old days, but now we have more tools at our disposal to determine what the diagnosis is. And so for this particular condition, we need to look at the chromosomes. And so just to do a bit of a recap of biology, our DNA, as was mentioned, is a nice helical structure, but there's just too much of it to stay in that structure in our cells. So it has to be tightly bound in proteins, wound up, put into scaffolding proteins, and eventually it forms what we know as a chromosome. And it's that format in which it's kept in the cells. This is a picture of the 23 pairs of chromosomes that we normally have, numbered 1 to 22, very imaginatively. But the last two are your sex chromosomes, X and Y if you're a male, or X and X if you're female. So in this CML condition, there has been a swapping of information between chromosome 9 and 22. So if we look at this slide now, there's a bit of extra information there, and it's missing a bit from there. Now that swapping of information causes a new gene to be made, which produces a new protein, which actually causes this condition. So why is that interesting? Well, first, it is interesting. Secondly, now that we know that's what causes CML, we can target it, and that's what has been done. So, whereas this condition before the year 2000 was quite a serious condition to have, only about 40% of people would survive beyond 5 years from diagnosis, it's now been transformed, and over 95% of people survive 5 years from diagnosis. And why? Because of a new drug called imatinib, which targets that particular protein and stops it from functioning. I was talking to a patient fairly recently, actually, who, when he was first diagnosed back in 1999, asked that question of the doctor, how long have I got? And he was told, average is about 3 years. But then he went into a trial, a clinical trial for what was then called STI 571, which became this drug. And he's now alive now, 14 years on, perfectly fine and happy. He has to come and see us every 3 months, which is a bit of a bind, but otherwise he's well. So for our patient James, he was started on one of the newer drugs called nilotinib, which is similar to this one, and he's doing very well. 3 months down the line, his blood count has normalised, his spleen, the lump in his tummy, has now settled down, and he's back at work, working all the hours under the sun. We can now use that protein level in the blood to monitor how he's doing. We can see how well this medication is working by measuring tiny microscopic amounts of that DNA and that protein. And should it increase, we can change medications now. We have more medications in the same line as imatinib that do the same job. So our next patient is Olive. She went to her GP as part of her annual medication review. As part of that review, she had a blood test taken. In herself, she felt very well. There was no problems, no health concerns. But the blood test revealed a high white cell count, and in particular, one of the types of white cells called lymphocytes. So she was referred to our outpatients department for review. Having seen these results, we asked for some more blood tests beforehand, just so we could get some more information. And then we looked at the blood film, the archaic bit of glass, under a microscope. And this was what it showed us. So these little sort of purple, reddish areas here are the red cell. The red cells. And these purple ones are, again, the white cells. Most of these are what we call lymphocytes, which are a type of white cell. But some of the features here, where these look like they've been smudged or smeared, are characteristic for a condition called chronic lymphocytic leukemia, or CLL. Now, again, that's where the story would have ended, but it isn't anymore. So we have much more sophisticated techniques for confirming the diagnosis. Now, as part of that, all cells in the body have proteins on their surface. They perform essential functions such as signaling between cells or import and export of nutrients and so on. But they can also be found on subsets of cells that help us determine what the cell is that's a problem. And they're all given different numbers called CD, cluster differentiation, which means that we can look for different patterns of these markers on cells and help us determine what is the problem. And we have a technique called flow cytometry, and that's a key part of our armoury now, where we can attach little lights, fluorescent markers to the cell surface, attaching directly to some of these known markers. And that helps us then determine what's going on. Then this gets passed through a laser beam. It takes only a matter of minutes to work. So the types of cells that are involved in CLL, chronic lymphocytic leukemia, they're B lymphocytes. And we know that B cells have CD19 on their surface, but they shouldn't have CD5. So in this particular one, you can see this large population of cells here has an abnormal marker on it, and that is characteristic of CLL. And these other panels also just look at other markers involved in that diagnosis. So when we went to see Olive in the outpatients department, we could already tell her, you've got this condition, and we could examine her and make the diagnosis and say, actually, with chronic lymphocytic leukemia, as you're well, what we often have to do is just watch you. We don't have to actually give chemotherapy of this condition or treatment. Often, it can just be treated by watching and waiting. We can also use some of those markers to try and predict how that condition will progress. One of the markers, called CD38, helps us determine whether this is a CLL that's likely to stay dormant and not be a problem, or is more likely to become a problem in the future. Now, whilst we were also in the outpatients department, our third patient came along, and her name was Eleanor. She's recently been put on warfarin for a blood clot that she developed in her leg. This isn't her leg, because I've been pointed out, it's a bit too hairy. So, but hopefully you'll be able to perceive that this is slightly larger diameter than that leg there. Possibly also that it's slightly redder, but maybe not with the coloration on here. Now, she's only 35 years old, and she's developed a blood clot. We all have an inherent tendency to develop blood clots that increases as we get older. But for most of us, even still, we need to have some sort of provoking factor that just triggers us to form a blood clot. Now, often that can be immobility, surgery, pregnancy, and that's why often when you go into hospital you get that nasty jab in your tummy to thin the blood, because at those times you're more prone for getting blood clots. But for some people, that tendency to form blood clots is greater, even without these provoking risk factors. And some people do have underlying genetic defects that cause this. Now, blood clots are a significant problem. It's estimated that 25,000 people a year in the UK die from blood clots, either in the legs or more usually going up towards the heart and the lungs. So it's a significant problem. We now have molecular genetic tests that can find out some, not all, but some of the defects that make people more prone to developing blood clots. And if you've had a blood clot when you're very young, that like in Eleanor's case was completely unprovoked, you may want to have more information about whether or not you're more prone to getting another blood clot in the future. And we can test for things such as factor V Leiden or prothrombin gene mutation and others, which help us make that decision and may impact on treatment in the future. Now, that takes us back to our last patient I should say really, who is Luke. He is 25 and he's coming to the A&E department because he is getting progressively more short of breath. It happened over the last 48 hours. About 3 days ago, he ate some fava beans. And so a blood test was performed and an archaic blood film, which was the start of the testing. These are the red cells, as mentioned before. They've had bites taken out of them and some what we call hemighosts, they're half not there. And that's classical of a condition called glucose 6-phosphate dehydrogenase deficiency, as was mentioned earlier on. Now, whereas that would have been sufficient before, we now have tests that look at the enzyme that causes this and the deficiency in that enzyme, the G6PD enzyme. And more than that, we can also now look at the genes to see if they have a defect affecting that. And why is that important? Well, already there are tests going on where that abnormal gene or a normal gene has been placed into mice and they've been able to produce the enzyme lifelong and prevent this condition. Now, in the future that may be able to be done in humans, but not yet. So we've looked at how modern technology can make use of your humble drop of blood. We can use it for diagnosing conditions such as CLL and CML. We can look at how it provides prognostic information to tell people how well they'll do with their condition, and how it will also predict the risk of further problems, such as in the blood clots. And more importantly, this is happening daily in a hospital near you. This isn't future technology, this is what we're using now. And more tests are constantly being developed, new technologies are constantly under development, and it's all from your blood. Thank you.
Well, it's 50 years since I was apprenticed to this trade, and things have changed a great deal. My first microscope wasn't quite the Leeuwenhoek one, but the two biggest presents I had in my life was a microscope with a folk camera on it, which was given to me with a Wellcome Trust. That's the most wonderful thing ever. And then a few years later, a quarter counter. And these are two of the best presents I think I've ever had. And I could play with these forever. But that's what I'm not here for today. Keep that last slide, actually, the interesting one you had of the glucose 6-phosphate. If you have a quick look at that, there's a rotten cells. People with that deficiency don't get malaria. And you can see why. Because the malaria parasites are very, very choosy. They're not going to get into rotten old cells like that, when there are plenty of beautiful ones which you've seen elsewhere. But enough of me at the moment. So could I ask the three speakers to come and join me? And they're prepared to ask questions. You could aim it at a particular speaker. It would certainly help. But the first thing you have to do is get my attention, and then you'll get a microphone. Lady in the middle here. Microphone come from.
Treatment in chronic myeloid leukemia had usually in the past had very bad prognosis. We use a chemotherapy and etc. A patient very quickly died. I would like to make inquiry how long is going this genotherapy, and if it's worldwide treatment or is only in England, is something very new.
Is this the CML?
Yes.
Yeah. Sorry.
So treatment for CML is now, that's a worldwide treatment now, been available since early 2000s. It is very expensive. It does cost, even if it's most basic, between £1,500 and £2,000 a month for treatment. But it's ongoing. The initial data from those who were in the trial and are now continuing to take the treatment has virtually levelled off. People are no longer, very rarely, dying from their CML. It's more from old age, from other problems and so on. So it has revolutionised that particular condition.
Can I add something to that? One of the things I get to do as the president is to write difficult questions for examination candidates. And one of the questions we've asked recently is discuss the relative merits of a stem cell transplant and Gleevec therapy for patients with chronic myeloid leukemia. Gleevec's not curative. It doesn't actually replace a missing gene. It blocks the activity of an abnormal gene, whereas a transplant gets rid of all the bad cells and replaces them with new normal cells. So these new therapies present us with dilemmas. It's actually quite difficult sometimes to decide whether someone should stay on lifelong drug treatment, during which new mutations could occur, and so the effectiveness of that blocking drug can be lost, or whether to suggest they should have a stem cell transplant and have a completely new set of blood-forming cells. That's, I think, still a dilemma, although most patients will opt for Gleevec.
And it's not worldwide.
Could I think when you were, oh sorry, all sorts of hands all over the place, so one here.
I think the answer might be probably rather more straightforward than the last one. Like many people, I suffer from type 2 diabetes. I get a blood check every year, and I'm told that the sugar level that is given by that test is the average for a 12-week period. I've never quite understood how a snapshot test can give an average for 12, and a 12-week period, not a 6-week period or an 18-week period.
Yeah, that's not that simple a question. I shall try to make the answer as simple as possible, but it's not measuring your glucose directly. What it's measuring is the amount of glucose that's been attached to your hemoglobin, interestingly, what we've been discussing. So it's called glycosylated hemoglobin A1C. And that builds up over a period. And there's a relatively well-known now, I think, well-accepted interval for testing, and that's 3 to 6 months. And that gives you a better overall picture of control than a snapshot glucose, which can go up and down like a yo-yo within a day. So it's a better test of long-term control of diabetes than just glucose measurements. It's called hemoglobin A1C.
Yes, I knew, but
Sorry, if you have the microphone.
The lifespan of a red cell.
Huh?
The lifespan of a red cell.
So there's I think it's leading on the other side, yes.
Well, the answer to your question is a red cell lasts that length of time.
Yeah.
So
There's a lady, Richard, on the other side, she put her hand up, yes. It always helps if you ask a question, right, and then you keep your hand up or wave it around, so like that she find you. So nothing is behind you.
Given that we are all from the same species, it does seem rather odd that somebody given the wrong blood can be killed. And it also seems odd that part of the O group seems to be a bit bog standard and can be given temporarily to a lot of other blood groups. Is there an explanation for this?
Okay, it seems like I've nudged for this one. You're absolutely right. It shouldn't be happening. Wrong blood shouldn't be going to the wrong patient. And if you look at our SHOT reports, which is our serious hazards of transfusion reports, which we have in the UK, most of that error is around manual processes. To actually give a blood sample, a blood transfusion to a patient, you have a blood sample taken, it's processed in the laboratory, the blood grouping is done, the transfusion unit is available, and then it has to be cross-checked with the patient and the patient's labelling before the blood is given. And the error is usually along that line. So it happens to be a technical or manual error or laboratory cross-matching, which is something that we do need to fix. IT doesn't fix everything, so it shouldn't be happening. The other question you said about O being given, as it's unexciting. Is it unexciting, but we have very low stocks of O. So we do tend to use O blood group because it doesn't have the antibodies and can be given, and we tend to save it for emergencies. There's a lot of blood required for a lot of the operative procedures we carry out, and so that's the safest blood to give in an emergency, but the ideal scenario is to get the right blood to the right person, and it's not necessarily the technology, it's the pathway for the blood which we do need to get right.
If I could just add to that, we had an appeal the other day for people to give their corneas from William Ayliffe, and this,
yes,
you won't do this, but on your behalf, please do donate your blood. It's life-saving for somebody, and particularly if you've got a rather unusual type, it can actually be a matter of life and death. So it's an easy thing to do to fill in the form. It's not too difficult to give in the blood, and it's much more difficult if you're cornea, but you only have to do that when it's gone. So 2 appeals in one. So, gentlemen here. No, the other side, sorry. And then behind that.
Thank you. Going back to that lady's question, in our species, how did the different blood groups evolve? And is there a geographical area for each type of blood group?
Well, I'm going to defer to you.
No,
Dr.
you're the
Prentice.
expert in transfusion.
I'm not the expert in transfusion. You start off with the younger generation and we'll correct him.
Thank you for that.
You're welcome.
The man who's most recently passed the
Passed his
exam is the
exam.
best one to answer these questions.
So, blood groups are inherent. Most things we develop antibodies to because we get exposed to them during our, to bacteria and viruses and so on. We get exposed to during our life, we develop antibodies against them. But blood groups are something we're born with. There's a few months' time when you're first born that you don't have any obvious blood group, but then you develop the blood group that you are going to have. And there are some geographical variations in that. I can't exactly remember which ones are which, but there are some slight geographical variations. But you are genetically predisposed to getting one blood group or the other, and then you will automatically develop antibodies against the blood group to which you are not, rather than because you've had exposure to another person's blood group.
Yeah.
Does that answer your question?
You'll gather that there aren't answers to every question. And blood grouping is still full of interesting questions. For example, we don't understand why some people lose their blood group and go from an A, B, or an AB to an O, or change their blood group. That's a very curious phenomenon that we've never fully explained, but it does happen.
I mean, it's the first few months of our lives before we're born that all these things are sorted out, whether we're going to be susceptible to disease, whether we're going to have a particular blood group or not, whether we're going to reject an organ at a later stage. Amazing things happen very early on. And I think it is a very difficult area to work with because humans are different from animals. And to work on fetuses of 1 to 2 weeks or 1 to 3 weeks is really not approved of. In fact, it's impossible. So there's nothing we want to know there. We know what the question is, but we know that the answers cannot be given. Have I got it right? Yeah.
Mm-hmm.
Thank you very much. Lady behind you.
Thank you. Regarding transfusions, do the vegetarian's blood, is it held in a different area to the meat eater's blood? Because if a patient is a vegetarian and they receive a blood from a meat eater, it may hinder progression or healing. I think it's a very important aspect we have to consider. Thank you.
If I could step forward to answer that. It's really the iron content that you're talking about in blood. And to be perfectly honest, yes, red meat has more iron than vegetables in general, but there are a whole host of vegetarians in the world and they don't have an issue with becoming iron deficient or giving blood. The only time I think you're not approved or you can't go ahead and donate blood is when you're iron deficient. And the reason for that is because it's a voluntary donation. If you're iron deficient and you donate a unit of blood, you become iron deplete. And for something that's a voluntary service to make you feel more unwell wouldn't be appropriate. But I don't think it makes any difference whether you're a vegetarian or a meat eater.
Well, is your question to do with mad cow disease? Is that what you're getting at?
Well, obviously, there's lots of discussions on that, but I feel, as a vegetarian, I wouldn't like the blood of a person who has meat.
Okay.
Understand me?
Yes.
It would hinder my progression.
Yes, there is no other therapeutic product, and blood transfusion is a therapeutic product that's so extensively tested. It's tested to exhaustion to try to exclude everything that can be identified. But that doesn't mean to say it will exclude everything that's potentially transmissible. So there's always a risk, and my generation of haematologists was taught a bag of blood is a biological time bomb. So we've spent, my career has been spent largely explaining to other doctors and other disciplines to transfuse as little as possible to minimize risk, but you'll never abolish risk of transmission of something.
There's a lady here, and then
some years ago I had very good blood actually.
so could you imagine? Sorry.
And I was wanting to donate, but they refused because they said I was too old. I mean, this was years ago, and I was quite upset because I was probably a lot healthier than people half my age.
Well, you never ask a lady how old she is. But there's a cut-off point, and that's blood transfusion service rules, and we can't do anything about it.
It's just that, as I say, I think it more depends on the health of the person concerned.
There's a second question from that gentleman. I think it's allowed. Yes.
In the 70s, I was reading that artificial blood. Heard no more about it, and I wondered what has it worked or anything, and what's the basic science behind it, please.
The US Armed Forces were desperately trying to produce an artificial blood substance at the time of Vietnam, which is why you were reading about it. They discovered that they could produce a substance that would carry oxygen and release it, but it didn't last very long, so it was never used in the field, as it were. And there's been no further research on that. What is happening now in the transfusion service is that the scientists are working on producing blood derived from stem cells. But again, the scaling up process is going to be very, very expensive. And for the moment, there's no foreseeable substitute for blood donors.
You're high enough for that. Yes, and then start to the front of that. Yes, that gentleman first, then I think it's the lady there, I can't see.
Bit of a prosaic question. You alluded to the potential and promise of molecular diagnostics, and that's very encouraging. But I just wonder about the potential workforce for the future of trainees. How do you secure that? When bear in mind that Sherwood, Bristol, and pathology reconfiguration, how do you secure...
You're not prosaic, you know very detailed information about how things can go wrong. Well, the answer is that as a college we're changing all our curricula. We've written a new curriculum for clinical scientists because this college is unique in that it has non-medical fellows and always has had. So we've written a new curriculum for trainees who are clinical scientists who are going to be purely molecular pathologists, have nothing to do with direct contact with patients. They'll work in molecular pathology laboratories. And we're taking bits from that curriculum and putting them into all our medical specialty curricula so that medical pathologists will understand the business of molecular pathology without having to do it. How do we ensure that the quality of molecular pathology investigation is good enough? I don't have an answer to that question yet. There's very little quality assurance or assessment goes on in molecular testing so far, and we need to develop that. We certainly need to develop that.
Lady there.
We have a joke in our family that those few of us who have group O blood have never actually been ill apart from breaking bones and things, and all the rest are pretty weak. Are we the dinosaurs, and hearing that blood had changed to O, are we the bog standard blood? And that's why we might not be very interesting, but we are useful temporarily.
I think it'll always be useful. Dinosaurs is quite hard to answer. There are a certain proportion of the population that are O, and there are certain that are A and B and AB positive. So I think it's just the genetics and the percentage in the population. There are very few that switch groups, and as Dr. Prentice says, we don't quite know how that switching happens. So whether group O people remain more healthy, it may be something that's genetic in your family, and long may it last. I'm not sure there's a general trend that I'm aware of, otherwise I'd switch group myself if I could. So I think it's just luck on the family side. The only other time that we switch blood groups is really in the bone marrow transplants that Dr. Prentice was talking about, when we're replacing someone's bone marrow with the blood, with another person's host bone marrow, and then the blood group switches. That's the only time we manipulate blood groups, but the rest, I'm afraid, is genetics and the environment. So long may you stay well with your group O blood.
Gentleman down here.
I understand if you're a carrier for sickle cell anemia, you actually have some protection against malaria. And I'm wondering if there's obviously a genetic advantage in that.
I think the best person to answer this is Professor Cox, actually.
Totally agree.
What the mechanism is and whether you could actually use.
Well, there are two parts. I alluded to that early on. The cells are sickled. It's not so easy for the parasite to get into them. And once they got inside, the various molecules they require are just not there. So it's just not a good place to be. Simple as that. Well, it's not as simple as that, because there are hundreds of people working on this particular topic. But that's a cartoon of basically what happens there.
Archie?
Did you want to add to it?
No,
No, the malaria parasites have got to have nice cells to live in,
Yes.
and
Yes.
sickle cells
You
just
saw
don't
the
provide
previous
that.
pictures, they draw them up.
I mean, the other advantage of being a biconcave disc is that you're very bendy. So you can get through spaces that a non-bendy cell can't get through. The sickle cell is literally, it looks like a sickle, and it's very rigid because all the hemoglobin S molecules, instead of being A, are all stacked up and don't bend. So parasites don't like that.
Can I come with one of your questions, your answer a moment ago? You were talking about the difficulty of getting standard diagnosis. To what extent is that due to different companies producing different kinds of equipment to do allegedly the same thing?
I'm assuming this is the molecular diagnostics.
Yes.
Is it?
Molecular diagnostics, yes.
Well, it's a complicated question. There is a regulatory authority, MHRA, which regulates all new equipment and all new tests introduced into the National Health Service. And it hasn't geared itself up yet to deal with molecular testing. It's a simple regulatory problem. It is a big issue, though.
One more question from the registrar.
Thank you very much for your talk and for focusing on certain objects. This is part of a collection series. So I just wondered, we'd like to thank you for having given us all the books with 50 objects, which is a kind of a virtual collection. But are there any other items in particular that you'd like to comment on? Or perhaps the way in which you've put these 50 objects together to form a sort of virtual collection of which we all now have a copy?
Well, I showed you a picture of the Guthrie card, which I think is one of the most precious gifts that individuals can make to society. They are taken from the heel of a child without consent, from a neonate without consent. And that's proved to be quite a difficult issue in some countries where there have been legal challenges on the grounds of data protection and confidentiality and consent. And in Southern Ireland, in Éire, for example, there is now a fierce debate raging about whether these cards should be destroyed because consent was never obtained when they were taken. We, as pathologists, feel this is such a valuable archive of material to go back to, because the cards are indestructible if you don't burn them. The blood is always there, the DNA is always in the blood spot. You can go back and track population trends in inherited diseases from these cards. I think that's probably the most exciting object in the book, curiously. It's a simple piece of paper with simple blood spots on it, which yields so much information with these new molecular technologies. That's my favourite.
I chose my three. I chose my three for my working life, but having looked back, I think for me it was the very small microscope that was produced by Van Leeuwenhoek.
Joe, do you want to add something to that?
I think, well, mine would be the glass slide. It's always been mentioned, I'm afraid. But,
yes.
Standardisation of the glass slide, so that all microscopes can use the same slide, same process, and the fact that it can give us so much information just from that one slide, and be the starting point for all the fancy tests we have. I think that's possibly my favourite.
So all this magic world here, people's wishes, when they've wiped the magic bowl or whatever it is, their good wishes come through. They're very simple ones, they're nice simple wishes, and I think that's what's happened all the way through. I was fascinated by this lecture. I knew obviously a bit of it, and it's been a rerun of my life, actually. And the bit you miss, of course, is that you see those beautiful blood slides. You try taking those with the humidity of 90 in the middle of Africa, with everyone breathing down your neck in a queue of about 150 children waiting to have their blood cells taken. And this is my only concern, discussion another time. This technology is wonderful, absolutely wonderful technology, but it cannot easily be translated to the sort of people that I used to be interested in the developing world. So what you really need now is miniaturization of some kind. The microscope you may have referred to as a MacArthur microscope, which was made a long time ago. It was about the size of a cigarette box, a very small box. It was a wonderful microscope, but it wasn't up to the standards required. So standards, in fact, are very, very important. So, well, I want to thank the three of you for a wonderful series of lectures. I've learned a lot, we've all learned a lot. I want to thank the questions as well, because my experience with the Gresham College is that the questions are always good, and the answers are always equally good. So we've got a very, very wide-ranging discussion. And we've been given a present. We've got a book to take home with us. And in the meantime, I want to thank all three of you very much, first of all, for giving up your wonderful time when you have to go back to Guy's and Thomas's, or Guy's, Thomas and King's, we can argue about that forever, or to Eastbourne, or to being chairman of the Royal College of Physicians, President of the Royal College of Physicians, which is a very, very important task indeed. And we do ask a lot of our NHS doctors, and we get there's a lot of stick about this at the moment, and people say, what are you doing? What are you doing about it? Well, now we know this is what it's what you are doing about it. So thank you very, very much indeed for giving up your valuable time and giving such a very interesting and informative series of talks. Thank you.